Are you going to the SEB meeting? Okay. No, I'm not there this year.
I got sucked into it somehow to co-organise a session. Otherwise, I wouldn't normally go to
Yeah.
SEB meetings.
But better Seville than Manchester or something.
Yes, I'm going for the whole week because I didn't know when our session was going to be. And then they just suddenly published the session. And not only is it on the last day, but I am speaking at 5:30 on the last day.
You're the closing keynote. It's all wheat. It's been live streamed.
Welcome, Christopher. Thanks for making the time available to come here to Rothamsted. We had some meetings already this morning and then now to come to dinner. So it'll be a seminar now. So just a brief background on Christopher. So Christopher is from Chile. He finished his university education agriculture as an agricultural engineer in 2001 and came an instructing associate professor at the Pontificio University in Chile. Then he moved to California in 2007, got his PhD there, so he was there from 2002 to 2007, and as a research assistant and postdoctoral fellow as well. And then after they moved to the UK in 2009, came to Cambridge, and then in the same year, I understand, joined in this center, which you all know in Norwich, and he got 10 in 2013. He's also has an honorary leadership and professorship of the University of East Anglia, and is affiliated scientist in Kenya.
Becca.
He has numerous awards and honors. He got the International Genome Sequencing Consortium Leadership Award in 2018 and the Royal Agricultural Society of England Research Medal in 2017. Also, he got from the John Innes Center the Knowledge Exchange and Commercialization Award and the President's Medal Award, Society of Experimental Botany from the UK 2014, and others. He also has numerous awards from doing his studies in Chile and also in UC Davis. Christopher has 83 publications with more than 6,000 citations and several patents as well. He has numerous BBSRC and IRIP, the International BPO Partnership grants, and also a LODA project on meiosis and wheat, and is also, of course, part of the Designing Future Wheat ISP. He's very active and very visible in the wheat research community, and so basically when you work on wheat, there's no escape, Christopher will be there one way or another. He's a really very popular speaker. He was invited to talk in 82 occasions in 18 countries. So we are really very honored to have you here in the UK, in home country at the moment. And we really look forward to your talk and update on what's going on in the region.
Thanks. Thanks, Sigrid, for the introduction and for the invitation. Thanks, Kara, as well. So today what I'd like to tell you a little bit about is the work we're doing in wheat and wheat genetics and genomics. And I always start with this picture in the field to remind ourselves that the objective of what we're doing is to get plants into the field. This is our experimental plot, but of course the idea is to get them into farmers' fields. And hopefully the fact that I'm ubiquitous is not a bad thing, I hope. But I think that for me, working on wheat, one of the big challenges that we have to face is polyploidy. The fact that wheat has multiple copies of its genomes. And polyploidy is not something that's just related to wheat. We see polyploidy across kingdoms of life. We see it in bacteria, in yeast, in frogs, fish, algae, and of course in flowers. In flowering plants, it's very important, right? 70% of flowering plants are polyploid. And many of the major crops in the world are polyploid as well, including ancient polyploid, who have all gone whole genome duplication events like the ones you see here, and also more recent polyploids like potato, wheat, cotton, and others, okay? So most of the plants that we work with are either old polyploids or they're actually current polyploids in their status. And I'll tell you more about wheat, of course, as the main focus of what we'll be discussing today. My approach is, as I said, as Sigrid said, I studied agronomy in Chile, then I did genetics, and now we do a lot of genomics and genetics and agronomy. We try to combine the different things. So I'll tell you things that we're trying to do, both trying to understand the genetics and the genomics, but also how that goes back to the field, which is kind of where the roots are and the importance of the work we're trying to do. And a few years ago, we published a review with Mike Bevan, and we were looking into kind of the resources that we would like to have in wheat. And hopefully this will show you the revolution that's happened in wheat genetics and genomics over the last 5 years, and many of this being led here by the UK. Now we have genome sequences that I'll tell more a little bit later on. We have natural allelic variation, so we have haplotype information. We have lots of data from epigenetics from Anthony Hall's lab, the tilling mutants that I'll tell later, expression data, a lot of SNP data, again, spearheaded by the UK, especially from Keith Edwards' lab in Bristol, networks, for example, with Netminer. And importantly, a lot of these resources, because they've been generated relatively recently, are all integrated into a common platform, which is Ensemble Plants, again, based in the UK. So the landscape in wheat genetics and genomics has really, really changed, and we were discussing that with the PhD students over lunch that, what before would have taken a whole PhD now takes, or a whole career perhaps, just to find a gene. Within a PhD, you can find a gene and start characterizing it. So it's really, it's very exciting what we can do now with these tools. And I'll try to tell you a little bit more about some of the tools and why we developed them. So of course, we have the wheat genome, which is great. There's lots of nice figures. But perhaps the more important thing is that we have the position of genes. So wheat, there's thousands of QTL studies. And now all of a sudden, we can put a lot of that data into perspective. We can see where the genes go. For example, when I'm selecting VRN1, what happens with this locus that affects cold tolerance and so on. So all of a sudden now we have all the genes in position. Importantly, this allowed us to also have a common nomenclature for many of the genes. Before, different groups would talk about different genes, different assemblies. Now we have a common framework that we can all speak from. And of course, we have a common reference to map the genes to. And this is very important because for so long, wheat was very disaggregated. The US would use one assembly, we'd use a different reference, Australia would use a different reference, the French would use a different reference. Now we have a common reference and we can start talking the same language for the first time. And of course, the wheat genome took 13 years to decipher, but just as quickly now 10 or even more new varieties have been sequenced as part of this 10+ pangenome project. So these varieties are sequenced and are actually available for the community to use. So from 13 years, within, let's say, 13 weeks of the publication of the genome, we had these other varieties already available. And again, the UK has played a leading role. These are just some of the varieties, but perhaps the most important thing I want to show is that this has not been done ad hoc. It's been coordinated so that some of the varieties that have been sequenced are really important for both UK breeders and also researchers. So two of these varieties, Cadenza and Kronos, are very important because these two represent parents of the tilling populations, which I'll explain later. Okay? And one of the big tools that we needed to do in terms of tilling was that, spearheaded by work that we did in the US, that Andy Phillips did here at Rothamsted, we were using tilling to characterize genes in wheat, and it was really great. But one of the problems we had is that people kept on saying, well, if we know that this gene does this in Arabidopsis, we'll know that it does this in wheat. But when you start looking at it, Arabidopsis compares to wheat as a platypus compares to us in the room, to humans. It's a quite big evolutionary leap to say that because it works in Arabidopsis, it works in wheat, right? Perhaps to put a caveat to that, Arabidopsis has been hugely informative for many hypothesis generations, but I think that now there's a really good reasons for working on Arabidopsis, but importantly, if you want to do stuff in wheat, you should actually be working on wheat itself, I think. And one of the tools that we needed to develop was develop resources that researchers and breeders could use. And that was one of the first things we said with Andy Phillips when we were in a trip in China my first year here. We said we need to develop this tilling research into something better. So as part of that, we started working with Andy and his population of the cadenza mutants and also the Kronos, which is durum wheat, population of wheat. And we took something very simple, just taking wheat with EMS, and basically you put them together and you get a population of plants that has random mutations across the genome. And that's very powerful. And for many years, we were doing tilling, which were basically we're taking the DNA and doing PCR of a single gene at a time. But that was relatively slow. So with Andy, we put a proposal together where we could actually take all the DNA of these plants and use exome capture. Okay, so we could actually take a molecular approach. We could make probes for all the protein coding genes of wheat, and we could capture the DNA and sequence it. And after sequencing it, we could actually identify all the mutations in the protein coding genes of wheat. Okay, so this slide is probably about 8 years of work, Andy, myself, and many others to actually get this research done. And this is very important because now we have a catalog of all the mutations in the wheat genome for this one variety, cadenza and Kronos. So currently, you can now go to ensemble plants, and this is how our wheat genome browser looks like. You have the gene at the top with the exons, and everything here is a mutation in one of those tilling lines that we've sequenced. So you can very rapidly go for your gene of interest, select a mutation like this one here, and this is the cadenza line 201, carries a mutation that leads to a premature stop codon. You press a button and you get the seeds from the seed store within a few days, and you have primers designed for that mutation so you can start doing your back crossing or your crossing very relatively easily. So it's really a transformation of what we can do in wheat, of going from months to identify a mutation to actually an afternoon of sitting in the computer and within a couple of days getting your mutants and start working on it straight away. And this has really been transformative. Over 7,000 mutants have been distributed over 327 orders over the last 2 years. So actually, most of the people that are working on wheat are using these mutations, and it's basically now become a staple of what we can do in wheat, thanks to the cadenza population developed by Andy, and also the Kronos population that we developed originally in Davis. So one of the other questions we had was, where is my favorite gene expressed? So it's a very simple question, but as we started having a common nomenclature, we started having the mutants, we wanted to know which gene do we need to target, which gene is expressed in the right place at the right time. And one of the things that was very frustrating here is that there were hundreds of data sets available in the public repositories, but we couldn't use them because they were mapped to different references, they were mapped with different programs, and the data was just sitting there. So in a way, for our own purposes, we started taking this data and analyzing it with respect to a common reference and making this available. And this grew into something bigger where we now have over 850 samples that we took from across developmental time courses of wheat. We sequenced 0 data. All the data was publicly available, and we used it within this data set. Okay? Just to illustrate the power, we were talking about this with the students over lunch, the power of reanalyzing data, of course, with quality control, but we're able to get all these samples and start analyzing them for the first time in common. And this started to reveal patterns of expression, so we could make browsers like this one here with EFP, very similar to what you have in Arabidopsis. So you could see this gene in particular is expressed in the roots, and you have all these different tissue time points that you can start to look. So now that initial question, where is my gene expressed? You can answer it, again, within minutes, you can answer that question in wheat. You don't need to make qPCR with hominid-specific primers for every specific copy. You can ask this question straight away. And we did different visualization interfaces. The current one is the one that we did in this expression.com browser with over 1,000 RNA-seq samples currently on the database as we speak. And importantly, this also allowed us to start doing some interesting biology as well. So I'll just talk a few slides on this genome of the wheat genes that were present in the A, B, and D genome. Roughly 70% were balanced, meaning that the 3 homologs were expressed roughly the same across most tissues of the plant. Okay, so in a way, they're quite boring. In a way, there's a certain expression, and the B and the D expressed roughly the same. And those genes tended to be genes that were expressed in all tissues and tended to be housekeeping genes. And then we started looking at some genes that were a little bit different, genes where one genome dominated over the others. For example, here that the B genome is much more highly expressed than the other 2. Or in this case, the A genome is suppressed with respect to the other two. And in those cases, many of these genes that correspond to about 30% of the genes, these tended to be more tissue-specific. They were expressed in fewer tissues at more specific time points. This also allowed us to look at how genes were changing their expression across tissues. So we started looking at many of the genes that were very balanced and that were also expressed across the whole plant, they were very stable across tissues. That means that in one tissue, they were expressed at a certain level, and all the tissues, they were expressed the same. They did not change their expression if you took leaf, grain, roots, whatever you took, the expression of the three was basically the same. And we found some really exciting genes, which are these genes here. These genes are very dynamic, meaning that in some cases, the B genome is very highly expressed. In other cases, the A genome is expressed. In other cases, the D genome is expressed. So they change a lot. The same genomic DNA changes expression depending on the tissue where it is. So these genes, when we looked at the promoters, there's a lot of interesting data in the promoters to suggest why that's driving it. So we were able to take, again, very data that was out there and start studying it and start understanding a little bit more about tissue specificity and how homologs, how the three genomes interrelate. And of course, now we're trying to link this up to biology. But it gives us a framework to start studying genes and start testing them if they're more stable or if they're very dynamic. And if they're dynamic, how does this influence breeding? So we did lots of networks. So this is stuff that you probably, if you've heard Kiwan talk, you'll see lots of these networks. So we did all sorts of different networks. But importantly, these networks are all really exciting, but they're very hard to use. So we've been working closely with Kiwan to be sure that we integrate this into NetMiner as part of designing future wheat. So actually, this data we've generated where we look for relationships between genes can actually be visualized in a context that's easier to look at. And then Kiwan has been instrumental in helping us achieve that. And again, by having a common reference, we're all speaking the same language. We're all speaking the same name of genes, and that's helped us tremendously. And of course, one of the things that we saw was that we needed to generate training tools for people to use this. The field changed so dramatically in 5 years that it was very hard to keep pace with what was the latest genome assembly, the latest gene models, the latest resource. It really was a complete generation change in terms of the resources we had. So as part of the things that we were doing, we thought that it was very important to get the training component so that anyone who wants to work on wheat, we lower the barriers and then can start asking these questions in wheat itself. And this page was actually done thinking of Arabidopsis, and we put it the other way around. We said, if I was to work in Arabidopsis, where would I start? I would have no idea how to grow Arabidopsis, how to cross Arabidopsis. So we started making sections about how to grow wheat, how to cross wheat, how to genotype wheat, how to make primers in wheat. Simple things, but there actually were barriers of entry for anyone who was not trained in a wheat lab. And that was very important because we're not going to solve the problems we have of food security and so on, just the same people working. We need new thoughts, and this was our attempt to try to get this. So now we have this page that you can go, and for example, if you look at functional studies, there'll be information about how to select mutants, how to do transgenics, populations that are available. Kostya was very kind and provided information on VIGs. So now all these resources that are available that if you're in the know, you kind of know about, we try to make them more explicit and easier for people to access. And again, this was within the Designing Future Wheat program. So I'll shift now and say, we've developed these fantastic resources, but now it's time to do biology. And that's, again, we're talking with the PhD students that for a long time, we were trained to do genetics and agronomy and so on, but now we actually need to start doing biology in a slightly different way because now we have the resources, we have the genes, we have the expression, and now we need to start putting genes together, pathways, and trying to understand how these things work in the field and with G/E interactions and so on. And we're really interested in looking at yield and specifically looking at grain size components. So this is a slide just showing the diversity of grain shapes and forms that you see in cereals. And in this case, we're very interested in looking how wheat, but also some of its relatives look, and also put this in context with rice, maize, and other cereal crops. So we had the challenge of trying to understand yield, and anyone who works in the field and knows about yield knows that it's one of the most complex traits because it integrates the whole life cycle of the plant. So in this case, we've been trying to look at, of course, you have the number of spikes, the number of spikelets, and you also have the number of grains. So these are the traditional yield components that you would be discussing. And when we started looking at yield, we hypothesized that to actually understand yield, we had to go one level down. We could not just stick with these components because all of these components have subcomponents within them. So if we wanted to understand the genetics of how yield was controlled, we needed to go down and understand the more basic principles of how these were affected. So in this case, we're trying to do a dissection. So instead of looking at number of grains and the grain size, we said, well, the grain weight can be controlled by the size of the carpel. Even before there's a grain that's formed, the size of the ovaries already tells you how big that grain can be. The size of the floral organs that envelop the flower also determine the grain size, the grain dimensions, the number of cells, and the size of these cells, and also the dynamics of grain development and how grain filling occurs. So actually, when we talk about 1,000 kernel weight or the grain or the weight of 1,000 grains, we're actually hiding all of these facts within that one component. So actually, we said if we want to understand yield, we need to go to this, and then we need to take one step further to understand any QTL that we have or any genomic region that affects 1,000 grain weight, which one of these traits is affecting, because we need to go down to that individual level. So again, as I mentioned, wheat is a polyploid, meaning that we have this very complex genome of 3 copies of every gene. So whenever I study a gene, usually we have an A, a B, and a D genome version, okay? Don't ask me about the C, but basically you have these 3 copies of the A, B, and D. So we have these copies, and they're relatively similar. Sometimes 70% of the cases, the expression is very similar. Sometimes it's different. And now you can start understanding this. But when we started thinking about polyploidy and yield, we started seeing this fact, that polyploidy, of course, buffers phenotypic variation. And this is a central issue that we have, that a lot of the variation we see is buffered by the fact that we have polyploidy. And I'll try to show you an example. In some cases, when you have dominance, when you have a mutation that's dominant over the other mutations, over the other homologs, sorry, over the 3 copies, with one mutant allele, you can go from the wild-type phenotype to the full mutant effect. And that's a dominant mutation, let's say, okay? In other cases, you have dosage effects, where you see, well, if I have 1 mutation, I might have 1/3, 2 mutations, 2/3, 3 mutations, the full phenotypic spectrum. Okay, so that's another type of variation we have. In other cases, we have non-additive dosage, where the single and double mutants might give you 5 or 10% of the phenotypic variation, and then the triple mutant gives you the full effect. And the worst case will be the functional redundancy, where single or double mutants look identical to the wild type, and it's only in the case of a triple mutant that you get the full phenotypic spectrum. And we have examples for all of these genes. So major genes like vernalization, photoperiod, the pH locus, resistance genes, these are, if you think about it, these are all the genes that have been very strongly selected because they're very strongly either semi-dominant or dominant. And you can see here that actually 1 mutant in VRN makes you spring, makes you insensitive, makes you resistant susceptible, gives you a certain type of spikelet architecture. Single mutations in 1 gene give you a dominant phenotype. These are QTLs, basically, okay? These are the QTLs that we've been trying to find. The grain protein content gene, which we cloned several years ago. Grain width, the red color gene. Subtle variation. You'd never get white or red. You get white, a little bit pink, more red, and red. You get this dosage effect. And you can see it very nicely there. And we believe that many of the QTLs that we work on are these dosage effects that are very hard to study, because it's not just that a QTL is basically underlined by many genes. And apart from that, the phenotypic effect is 1/3, because you're kind of splitting the phenotypic effect by 1/3 because of this homology. And then these genes here are genes that you would never have cloned in wheat directly, because there's no phenotypic effect to clone them with. You only find these genes by reverse genetic, by taking the waxy gene from maize, making the mutations in wheat, and you see it. The powdery mildew resistant, start branching enzyme. These are all genes that you can only find if you know the homolog or the gene in another species, and then you test it in wheat. So therefore, we hypothesize that there's a lot of variation, that we selected this variation, but all of this variation here has remained hidden variation that we have not been able to select through traditional means. And the exciting part about genomics now is that for the first time, we have all the genes, so we can start now doing these tools. We have the TILI mutants, we have CRISPR, we have the expression, and all of a sudden we can start unlocking this hidden variation that has basically never been selected or never has been used in breeding until now. And that's the exciting part about why genomics in a polyploid is especially relevant. And I'll show you an example with grain size, again, because this is where we're thinking. If you look at all the published QTL studies in rice compared to wheat, okay, there's not that the wheat people are really bad at doing this stuff. It's just that biologically, this is the effect we get. If you look at QTL papers in wheat, 1,000 grain weight or grain length or grain width, that's the phenotype. If you get 5%, you're really happy with a QTL that gives you a 5% difference with respect to the other parent. In rice, that QTL, you wouldn't even publish it. Basically, you need to have 20, 25% is an average phenotypic effect of a QTL in rice. And the ones in wheat, they wouldn't even be published, right? So that's the difference that we're seeing because, of course, rice is a diploid. And these are the phenotypic effect of single mutations in length and width compared to the ones we have here. Okay? So we hypothesize, is this because the genes controlling grain size in rice are completely different than the ones in wheat? Or is it because of polyploidy that's buffering this effect? So what we're seeing, these QTLs are that dosage effect. But that means that we hypothesize, therefore, that if we were able to combine mutations across all 3 homologs, we might be able to see higher variation. That's the hypothesis that we put forward. And we took one of these genes called GW2, where the single mutation was able to increase grain size by 6%, similar to what we show here. But then when we made a double mutant and a triple mutant, now we were able to increase grain size by 21%, very similar to rice. So we hypothesize that much of the QTLs that we've been looking at in wheat are basically recessive traits, and these are recessive so that when you can combine mutations across all 3 homologs, you open up previously hidden variation. And it doesn't mean that we want to create plants that have 21% bigger grain. It means that for the first time, breeders can actually select this and breed with it, whereas before, they were selecting this, and we're only able to breed with that sort of variation in natural alleles. Okay? And I'll show you some yield data now for this mutant there. So we took that single mutant in the first GW2 gene that had the 6% effect on 1,000 kernel weight, did a lot of backcrossing to make up lines that were very similar, but were different in this mutation. And now we have a nice and fancy experimental station. So perhaps for you guys here, this is kind of obvious, but at John Innes, agronomy and field trials are not obvious. So it's been 10 years of saying, this is really important, the field is important. And I think it's really great that it's been recognized that actually buying a tractor and buying a field station and constructing a field station is equally valid as getting the latest SEM and TM and all these microscopes. So I think that for the first time, we have a recognition that what we do is a valid way of going about it, and the field is really important. So I'm just showing this off because this week it's going to be officially open, and we're really proud of now kind of moving into the field and with no shame, right? It's fantastic to be doing stuff in the field, and I think we're now very proud of that. And we have our yield trials where we've taken these near-isogenic lines and we put them in the field with 6-meter-square plots. To look at yield, we do 10 replications because of the power that we need to detect these subtle differences. And I'm going to show you now a slide that has a little bit of data on it. Okay? So these are different years in different locations, and this is another year. This is the mutant that has the 6% larger grain, and this is the wild type. And you can see with different replications that although we get the effect on 1,000 kernel weight, this does not translate into yield. Okay? So this is the tricky part about working with yield, that you can affect one component, but the plant fights back. In this case, it's fighting back because the number of seeds per spike is dropping. So basically, the plant says, OK, I'll make larger grains, but therefore, I'll make fewer grains. And by that, it's compensating, and there's no yield effect of the single mutant. And this is what breeders are normally dealing with, these small effect genes and so on. So now what we want to check is what happens when we put the triple mutants in the field. And now we have for the first time the triple mutants, and we want to see if we can break these very strong correlations that we always get, that one goes up, one goes down. If we actually kind of really push the system to make grains that are much bigger, will the plant be able to compensate or not? Again, it might not work, but for the first time we can purposely make plants that are bigger grains or much bigger grains than we could before, and we can start doing the physiology and so on. So we'll see if this works, and then we'll continue fighting. We'll need to combine it with other traits we know. But again, for the first time, we can purposely do it rather than just hoping that something will be bigger. We can engineer plants that have bigger grains, and now we're going to see how these work. Very interestingly, of course, when you make bigger grains, you can also be affecting quality, and that's something that we didn't want to compromise. I started my work on wheat looking at grain protein content and micronutrient content. That's something very close to my heart. So we always looked at these, and this is a very interesting mutant because although conventional wisdom will tell you if you make lines in breeding, you have lines with bigger grains, those lines will have lower protein. This is this very established correlation, but it's not causation. And when you start getting to the genes, in this case, these lines here that have bigger grains, not just us, but also a publication from China shows that these lines actually have more protein content than their counterparts. Why? We don't know. But for the first time, we can start asking these questions. So we're starting to break these correlations. And as part of that, we also worked with Jannica Balk and others at John Innes to try to see if we could do ways to increase zinc and especially iron content. And iron is one of the things that has been very difficult to increase in wheat grains. We can increase it 5 or 10%, but we've kind of reached a limit. So with Janica, we came with a different approach, in this case, a transgenic approach. Using a high molecular weight glutenin promoter that was developed or characterized here at Rothamsted by Peter Shuri's group, we hooked it up in front of this vacuolar transporter of wheat. So it's a wheat vacuolar transporter that pumps iron into the vacuole, but it's not expressed in the grain. So we took the high molecular weight glutenin and we said, how about if we express the pump in the endosperm? Can we actually load more iron into the vacuoles in the endosperm? And you can see here that this is the control line, and here we're staining for blue for the iron. And with 1 copy, you can see there's more iron, and with 20 copies, there's much more. So we were able to increase the iron quite substantially in the white part. And then we sent some of these grains to Peter Shuri. He did the roller mills and so on, and we were able to see now that in the mutant, or let's say the transgenic lines, we have a much higher iron content in the transgenic lines in these initial breaks, so the white flour, but not in the whole meal. So there's equal amount of iron in the grain, but the iron, instead of going to the outside, to the bran, where it's usually taken off, when we get white flour, it actually stays in the white endosperm. So we can now make white flour with high amounts of iron. And we're combining these transgenic lines with the big grain size. We're making these combinations to start seeing how these interact. And importantly, we've also taken this to the field. And again, in a way, following on from what Rothamsted has done of taking the biotechnology into the field, we have our first transgenic field trials at John Innes under this consent. And people say, oh, it's quite horrible and so on. It hasn't been that horrible so far, at least. No protest so far. We've done a lot of media communication and trying to explain why we're doing what we're doing, how we're doing it. But I think we're quite happy with the experience so far. And I think it's important that we continue as a community to push transgenic trials into the field to test them, because I think that now it's becoming every time more normal. Again, thanks in great part with what happened here at Rothamsted with wheat, the transgenics. In a way, for us, it was very normal to take that next step, and we haven't had any complaints so far, only very positive feedback. Okay, so I'll take you back now to the grain size story. So we had this gene that we know affects grain size, which I mentioned, this GW2. But we also were working on QTLs. This is natural variation that we find in UK germplasm. So this is a cross between 2 UK varieties. And we found a QTL on chromosome 5A that affected grain size. So it affected 1,000 kernel weight. And these are the positive lines. So these are near isogenic lines. So these are lines, and every year they have increased 1,000 grain weight. As I mentioned before, we wanted to understand if this was due by length or width. What was the basis of this? And we see that if we look at the grain length, every year we increase the grain length by about 4%, a very stable effect. And when we look at the grain width, we see that we increase grain width by 2 or 1%. So this is kind of a minor effect. But actually, when you compare the 1,000 kernel weight and the grain width data, there's a very strong correlation. So for the first time, we started looking at it and saying, well, is the effect caused by the width of the grain or by the length? We always get this 4% length effect, but it seems that the width is actually very highly correlated with the grain size. So what's going on here? And I'll tell you what we think is going on. And just to say that this is a lot of work coming from Jemima Brinton, a PhD student, now a postdoc in the lab, and James Simmons, who's the RA, who has the long-term vision of developing all this germplasm and the field. So by having near isogenic lines, not only can we start looking at these quality traits or these grain size traits more in detail, but it also allows us to do a little bit of physiology in terms of growth curves and comparing them. So we could take, for example, lines from ovary to 0 days across anthesis, after anthesis, right? So these are all days after anthesis when the grain is forming. And we could start comparing at what point do these lines separate in their size effects. So you can see that these lines for grain length, they're the same before anthesis and up to 5 days after anthesis, but then soon after they start separating. So about this time here, the QTL starts having an effect. But for grain width, you can see it seems to be a secondary effect. It's an effect that comes on later. So there's no difference here, and only at the end do we have a width effect. When we take these lines, we're also able to see if it's cell division or cell number. And we find that it's cell expansion. So we have equal number of cells in the 2 grains, but the grains that are longer have cells that are longer. So we have now trying to get to a mechanism that this is not due to by cell division, but it's actually cell elongation that's causing the grains to be longer, which causes the grains to be heavier, which causes the QTL for 1,000 kernel weight and for yield. So we started now doing combinations of these near isogenic lines. So I showed you before, when we manipulate one effect, perhaps we don't always get the yield effect. What happens if we start combining something that affects width with length? So in this case, this grain weight QTL that I showed you before affects cell expansion, and this is 5A. And we combine it with this gene here, that's a gene that affects width, that affects cell division, and affects not grain development, but affects ovary size. So both are QTLs for 1,000 kernel weight, but they affect 1,000 kernel weight in completely different ways and in different times. Before anthesis, after anthesis, cell division, cell expansion. So again, we hypothesized that if we combine these two, we perhaps would have more stable effects on yield because you would have two independent mechanisms of increasing your grain size. So again, we go with these lines into the field and yield plots, as I mentioned before, and we start looking at interactions, trying to get a little bit at the G by E. Okay? So we're looking at genetic background, environment, also water status, and also nitrogen fertilization. And we did a big factorial experiment to test these traits. This is how the field looks like. So this is last year, 23rd of June, the irrigated trial and the non-irrigated. In this case, that's normal nitrogen and high nitrogen. And if you remember, last year was a very dry year. So just a couple of weeks later from this, you can see now that the non-irrigated is really suffering. It's really senesced and completed its grain filling versus the irrigated trial and the high nitrogen that has even more. So we're very happy that we had a very good year to test the hypothesis that we were looking at. So when we started analyzing the data, this is the data all combined before we start doing the factorial. Okay, and we start seeing that, first of all, we have no interaction of nitrogen. So nitrogen does not change the effects of these genes. So in this case, both genes affect 1,000 grain weight. Together, they affect more. And both genes affect yield, but not significantly. But together, they affect yield significantly. The 5A affects length. The 6A affects width primarily, right? So each QTL affects it by itself, but together, they're actually much better than either one on their own. Okay, but now let's look at those interactions. The 6A QTL does not interact with water. So it doesn't matter if you're irrigated or non-irrigated. The 6A effect is very stable. It does the same thing. It doesn't care. But 5A has a very nice interaction with water. Okay? So we have interaction of the 5A QTL with water both for 1,000 grain weight, for width, and it's not significant, but it's less than 10 for yield. Okay? And I'll show you the data so you can see what I mean by this. So in this case, in one genotype, we have that with no irrigation or with irrigation, the length effect is completely consistent. It doesn't matter if the plant has water or doesn't have water. The length of that grain is always 5.5, 5.7% longer if you have the QTL. But look what happens now with the width. The width, now suddenly, that small effect that I showed you before now becomes a bigger effect. You have this 2.4%. That leads to bigger 1,000 kernel weight, and that leads to higher yield. Okay? In the other genetic background, the same. And this is a paragon background, two different genotypes. No irrigation, there's no effect on the length, on water. Water does not interact. It doesn't matter. The length is fixed. But if you have water, you make a wider grain, which makes it a heavier grain and leads to increase in yield. Okay? So now this was very exciting for us because for the first time we're seeing what we think is a G by E interaction like really before our eyes. And I'll explain what we mean by that. There's a genetic effect, which there's an early effect of this 5A QTL on the length. So it leads cell expansion, leads to consistently longer grains. Be there water at the end or no water or fertilizer, no fertilizer, the length effect is there. It's always giving you that length effect. But that length effect gives you the potential to fill more towards the end of grain filling. Therefore, if the environment, this determines that the potential is realized. So if you have more water in that environment towards the end, the longer grains can fill more, then the increase in 1,000 kernel weight and yield is higher. So you make a longer grain, and if the environment allows you, you make a wider grain. And therefore, the magnitude of the effect on yield is given by the width effect, because you're always going to be longer, but the environment tells you how wide you are. How wide you are tells you how heavy you are, and that's correlated with your magnitude on yield. So in a way, this is G by E. Genetics is always there. The environment gives you the magnitude of that yield effect depending on the year. Okay? And now just to show you the data from the different years, this width effect always comes on at the end, and it varies year on year. And basically, the length effect is always there, and the width is the G by E. Okay? So this would tell you that this QTL would be great in certain environments that have water towards the end of grain filling, more so than those environments that are quite dry towards the end. Okay? So this is G by E, and we're actually, for the first time, understanding it. So we're very excited. And we put these QTLs also in different backgrounds. So currently, Many of these QTLs are also in CIMMYT backgrounds. So we've done backcrossing to put them into different varieties of CIMMYT. So these are the different NILs that are currently growing in Obregon, and this is all part of the International Wheat Yield Partnership that Sigrid mentioned at the beginning. Okay, so we're trying to understand yield from the field, how it interacts, but at the same time we want to understand what are the genes that are controlling these traits. So our original neo-isogenic lines have a really big introgression. It's about this big, so actually it's more than, this is the QTL interval, and look at the size of that. That's still the size of the rice genome. It's a huge chunk of DNA, and we can fine map it to about 75 megabases, roughly a little bit smaller than the Arabidopsis genome. So it's a huge chunk of DNA, but now it's all changed now, right? Because now, before this, okay, we have a QTL, we have a chunk of DNA. What do we do now? The breeders can breed, maybe, whatever. But now we have all the genes, right? We have the genome sequence. Now we know there's 461 genes in this reference cultivar in Chinese Spring. We know that, right? But importantly, we can now take advantage of the 10 genome project. And that's very important because it doesn't matter that Chinese Spring has these genes. What do UK varieties have? Because that's our original population. So we looked in Charger and Badger, and we took all the SNPs that Keith Edwards' lab had developed across these big projects, and we found that we had about 100 SNPs across the region. There's just a representation, but we had about 100 SNPs across these 461 genes. And we tried to analyze them. Do any of these genes or SNPs change an amino acid of the gene, and could that be the gene, and so on. But actually, we said, well, if we start looking at the genome data, Charger is related to one of the sequence varieties, and Badger is also the grandparent of one of the sequence varieties. So when we looked at that, we said, well, if we looked at all the SNPs in Charger, which is the parent of the low parent, and we compare it with Claire, which is one of the sequence UK varieties, they're actually identical across the region. If we compare Badger, which has the positive allele, and we compare it with Cadenza, the parent of the Tilling population, but also a line that's been fully genome sequenced, we have exactly the same sequence, the same SNPs. So therefore, we said, well, instead of looking at variation at these 100 SNPs, from one afternoon to another, we now know all the sequence between this marker and that marker in Claire. We know all the sequence in Cadenza, and we can now compare them directly. So we took those 461 genes, compared their sequences, and basically we found a few genes that had mutations that could be of interest in one afternoon. And that's the rate of change that we can now do. It's remarkable what we can now do. This would have been a whole career. This would have been my life. Hopefully this will be a nice postdoc project now. It's really, really remarkable what we can do. And of course, we can now use our resources. We developed these resources to do biology, not just for the sake of it. So we could take that, the EFP browser, look at the expression of all these genes and those genes that had polymorphisms. We could look at model species, Arabidopsis. What do these genes do in Arabidopsis? We had RNA-seq data from these neo-isogenic lines at the right time, so we could look which genes are differentially expressed. And then we could combine this with the Tilling population and find mutations, in this case in Kronos, which is our population of tetraploid wheat, find mutations in the A and B, make an F1, self-pollinate that, and basically now phenotype the F2, double mutant, double wild type. Are they different? And we can do this within 10 months. We can test this hypothesis. Again, this would have been many months or many years before in wheat. Now we can do it in 10 months. And again, all of this is being done now with speed breeding, developed by Lee, by Amy, and others here in the UK, to actually implement speed breeding so we could actually do this process much faster, the first 2 generations at least, and then phenotype in normal glasshouse. And now we can start seeing, of those genes that have variation in those sequence varieties, do any of them have any interesting phenotype? And then 4 genes, we tested 5 genes. 4 genes, no phenotypic effect, and one of them did have an effect. So this is one of the genes that we worked on. So this is 1,000 grain weight in the field now. For one of the lines, it has a certain distribution. The mutant in the A genome, the mutant in the B genome, and then the double mutant, we get a much, much heavier grain. Again, dosage effects in a polyploid, single mutants give you a dosage, double mutant reveals the full phenotypic variation that can be uncovered in that single loci. This variation was due to grain length and also to grain width, because with these plants we really water them to be sure that we were able to maximize that effect. Okay? And now we have this candidate gene which we're evaluating more. And when we looked at the distribution in terms of the growth curves of the grains, just like we did before, you can see that we phenocopy. We have the grain length effect early on during grain development, so about 10 days we start seeing the first differences there, but then the width effect only comes on at the last stage. So basically, we've taken now independent lines in a chronos, in a durum background, made a double mutant, and when we study how those grains develop, we phenocopy the growth curves that we had before in that natural variation. So we're really excited with this. It's a gene of unknown function, so now we're basically back to square one, but we're really excited because now at least we have a handle. We think this is the gene, okay? It might be other genes that also affect yield, but we think this is one of the genes that's controlling yield. And again, this is something that would not have been possible without the tools that we've developed. Okay? And just to show that we've also taken that mutation, which is present in UK varieties. It's not that exciting, but it's... Okay, so, well, it kind of is, but anyway. So, but just to show you this is the difficulty of working with yield, okay? So if we take the breeding program from Limagran, n equals 100, you cannot say the number, but it's a big number, you can imagine. We genotype it with this marker. This QTL is a QTL that they had not found before because this SNP was not a SNP in the data set, and there was no SNP in LD. So basically, there were no way of tagging this SNP. So we can see that we increased 1,000 kernel weight in the 2018 data. When you have it, this is hundreds or even thousands of data. It's a large data set. And then this is taking into account all their lines with all the different variation. If you have this line, basically you increased yield by 0.35. So you say, well, that's nothing. Simon Berry and Ed Flatman were like, wow, this is, having a marker that controls yield just plain blank like that is extremely exciting, okay? So now, of course, we need to understand, this is just a whole data set. We haven't blocked it for population. There's lots of things we need to do, but just shows that, again, we're trying to get this information back to the breeders to understand how this is actually relevant within UK breeding programs. So with that, just some final thoughts, that the genomic tools that we developed, the idea is to do biology with them, right? Not just to develop them for the sake of it, but hopefully I exemplified how we can use these different tools to understand the biology and how that accelerates our understanding of biology in a polyploid. Polyploidy hides extremely useful phenotypic variation, and I think that's why it's so exciting that what we can do in wheat, especially for now all the young people, that now all of this is your job now, understanding that hidden variation, right? And that's the exciting part now. And for breeders, they're excited. And of course, making triple mutants is not easy, right? It's not the best thing for breeding. So we also want to understand how we can make dominant mutations. That's one of the things that we're really interested in. There's really cool papers in tomato of how they've done CRISPR and promoters to make variation. So we're exploring that as an avenue of how we can generate dominant mutations that can give us this phenotype, basically in those genes that would be hidden before. We think that combining complementary modes of action is a rational way of understanding yield. And then cloning yield component QTL is possible. Still difficult, still requires a lot of time and energy, but it is possible. I think that that's going to be the new wave in wheat, that we can understand QTLs that we couldn't do before. And of course, all of this is great, but it needs to go back to the field. So with that, I'd be happy to take any questions, just to thank all the people in the lab, Philippa now at Birmingham and Olu, and just to remind that it's not just about wheat diversity, but also about human diversity. Thank you very much.